And this is going to be Chapter 3, Understanding EKGs, and this is going to cover basic electrophysiology. Okay, and the objectives of this chapter are going to be name the two basic myocardial cell groups, describe the function of each myocardial cell group, discuss the four primary properties of cardiac cells, list the three major electrolytes that affect cardiac function, describe the movement of ions, describe cardiac depolarization, describe cardiac repolarization, define the refractory period, describe the absolute refractory period, describe the relative refractory period. First thing we're going to talk about here is basic cell groups. You have myocardial working cells, and they're responsible for generating the physical contraction of the heart cells. So whenever we talk about myocardial working cells, the actual contraction itself, these are done by the myocardial working cells. They make up a thin muscular layer of the atrial walls and the thick muscular layer of the ventricular walls. Primary functions are both contraction and relaxation. Physical contraction of the myocardial tissue actually generates blood flow. So whenever the heart is contracting, you get blood flowing out either into the pulmonary trunk or out to the body. Specialized pacemaker cells. They don't have the ability to contract, but they are responsible for controlling the rate and rhythm by coordinating regular depolarization found in the electrical conduction system of the heart. You have two types of cell groups. One is myocardial working cells, and the other one is pacemaker cells. So again, back on the previous slide, one type is myocardial working cells, and these are going to be the ones that are actually contracting and doing the work. That's why they're called working cells. The other ones are going to be pacemaker cells, and these guys have the ability to put the electrical conduction system into play or actually stimulate the myocardial working cells. Primary function is to generation and conduction of electrical impulses, and they have the ability to create electrical impulses without being stimulated by a nerve. So these things are self-generating, they're running all the time. The pacemaker cells themselves can run without any stimulation from a nerve whatsoever. Even though they are innervated by sympathetic and parasympathetic receptors, they do not require those to actually run. So these things have their own ability to generate the electrical impulse all by themselves. Threshold. I like to think of threshold as the edge of the cliff, a point at which the stimulus will produce a cell's response. So whenever we generate enough electricity to occur and we're at the edge of the cliff, once we've achieved threshold, it's pretty much downhill from there. And then the all or none phenomena. If the stimulation is strong enough, all cardiac cells are to reach threshold, all cells will respond to this stimulus or none will respond. So what this means is that in the all or none phenomena, either all the cells are going to respond or none of them will. This isn't going to give us a half product of any sort. It will give us a full product every time. Either the heart is going to beat or it's not going to beat. There's not going to be anywhere in between. Four characteristics, or the primary cardiac cell characteristics, and they possess four primary cell characteristics. One is contractility, also referred to as rhythmicity. The ability of the cardiac cells to shorten and cause cardiac muscle contraction in response to an electrical stimulus. 2. Automaticity, ability of the cardiac pacemaker cells to spontaneously generate electrical impulses without external stimulation. 3 is excitability, ability of the cardiac cells to respond to electrical stimulus, also referred to as irritability. And then conductivity, the ability of the cardiac cells to receive an electrical stimulus and then to transmit the stimulus to other cardiac cells so that they function as a unit. Alright, quick review on this again. Four basic cell characteristics. They have the ability to contract. This is a mechanical function. They all have automaticity. The cardiac pacemaker cells can kick out an electrical impulse all by themselves without any kind of external stimulus. They are excitable. If you actually could touch the heart just from the touch alone would beat, or would try to form a beat from that location. So the heart is very excitable. That's why we get things like ventricular tachycardia and ventricular fibrillation, is because the heart itself, or the myocardial tissue itself, is excitable, and then it has the ability to conduct. In my live lectures, I tell people an explanation of this is all muscle cells have the ability to conduct electricity. The body itself is very conductive, so the heart is even more conductive. That's why we use defibrillation. That's why NAED works. The electrical stimulus will conduct through the tissues. The defibrillation from the AED or the manual defibrillator just causes a depolarization, or causes the heart to contract whenever the artificial electrical stimulus is given. Primary pacemaker cell characteristics again here, and this is pretty much to cover the location. Automaticity occurs in the SA node, the AV node, and the Purkinje network, and this is down in the ventricular muscle itself. Excitability pretty much occurs in all the cells. Conductivity, again, all the cells. And then contractility. The pacemaker cells didn't have the ability to contract and relax, but the myocardial muscle cells sure did. So the first three are an electrical function, and the last one on the list is mechanical function, which is contractility. Major electrolytes that affect cardiac function. An electrolyte is a substance whose molecules dissociate into charged components or ions. They produce a positive cation or a negative anion when placed in water. Major cations that affect cardiac function are going to be potassium, which is pretty much inside of the cell, and sodium, which is pretty much outside of the cell. And these are vital in the ability of the heart to actually depolarize or contract or cell shorten, whichever term you'd like to apply to it. But the ability of the heart to contract itself is essentially driven off of something called the sodium-potassium pump. Calcium is a pretty important role as well. From A and P, you should remember a cardiac cycle looking something like this. Up front here, you had the sodium-potassium pump, and then you had an influx of calcium. Calcium, pretty much, calcium extends the length of the contraction. That's why we have an organ the size of a fist that can actually produce cardiac output for a whole organism or the body. Calcium is essential. It kicks off the contractions for in the muscular environment. It elongates or slows down the contraction. Very, very important to understand calcium's role. Movement of ions. Ionic difference on the two sides. Potassium is pretty much inside the cell. The sodium is pretty much outside the cell. This works and is allowed to move inside of the cell by active transport, or if we had a cell wall, the sodium-potassium pump would be here. Potassium, this is going to be inside the cell. This is going to be outside the cell. And sodium would be there. To change the polarity so that the contraction or the shortening of this will actually work, this pump is activated, which brings sodium in and potassium out. During depolarization, this occurs. Calcium influx as well into the environment and elongates the contraction. The resting state or polarized state, the inside of the cell is generally negative relative to the outside of the cell. And there's a better picture here in just a second. When impulse develops and spreads throughout the myocardium, changes occur in the heart muscle fibers. During cardiac depolarization or contraction, Sodium ions rush into the cell, changing the interior of the cell to a positive after the cell is stimulated. During cardiac repolarization, sodium ion returns outside of the cell, and this is relax. Sodium ions return to outside the cell, and potassium returns to the inside of the cell. So, again with my cell wall here, we have a sodium-potassium pump. Potassium is on the inside, and sodium is on the out. This is flipped during depolarization, so that potassium is on the outside and sodium is on the inside. Now, whenever repolarization occurs, it again is flipped through the sodium-potassium pump. So depolarization, sodium rushes into the cell, potassium out. Repolarization, sodium goes back outside of the cell and potassium comes back in. And if you can imagine every one of these having a cell wall. This is inside, this is out. This is a state of readiness, but this is what's going to occur. And then whenever it repolarizes or recovers, potassium on the outside will come back into the cell, and the sodium on the inside will go back out to the outside cellular environment. Terms to remember: resting membrane potential. State of the cardiac cell, the inside is negative and the outside is positive. So in resting, inside is negative and the outside is positive. Action potential. Change in polarity produces a change in the cell's electrical charge caused by stimulation of the myocardial cells. Syncytium. Cardiac muscle cells, groups that are connected together and function as a unit. If you'll remember back from A&P, they're grouped because they have special little parts to them. And those parts are considered intercalated discs, which allows the muscle cells to move electricity, kind of like pond skipping, so that this entire area shortens all at the same time. Polarized state. Resting state of the cardiac cell. Inside the cell is electrically negative relative to the outside, which is positive. Refractory periods. Attempts to ensure muscle is totally relaxed before another action potential or depolarization can be initiated. Atrial muscle equals 0 to 15 seconds. Ventricular muscle equals 0.25 to 0.3 seconds. Period of rest is referred to as cardiac repolarization. There are two stages, and this goes back to this all or none. So whenever this occurs, this is a Q wave, this is an R wave, and this is an S wave. And we'll learn these here in the next couple slide sets. The QRS signifies ventricular depolarization. So the ventricles themselves are contracting during this time frame. The T wave, which is this one here, identifies the repolarization of the ventricles, which occurred here. So during this time frame that we're seeing here, this is considered the absolute refractory period. And this goes back to our all or none theory, that this zone that we're looking at is absolutely refractory to any outside stimulus, cannot spontaneously depolarize at all. This right here, this little zone, is considered the relative refractory period. And I want to explain this. This is the T wave that we're looking at here. And this is halfway through the repolarization, repolarization of the ventricles. So if the stimulus was strong enough in this area, like say your patient had a PVC, it could generate its own beat or be generated from the location that the PVC was from. What this will give us is this was called an R-on-T phenomena, and this will give us VFib or something that looks like shark teeth, which is VTAC. Either way, relative refractory period. This is relatively refractory, but if the stimulus was strong enough, it could affect it. Summary. You now know the two basic myocardial cell groups. One, which are myocardial working cells. They have the ability to shorten. And two, which are specialized pacemaker cells. These pacemaker cells are located throughout the sections of the heart. And we'll learn in the next slide set that each one of them has a specific rate that we're going to look at. And they make up the electrical conduction system and how they function to cause each cardiac cycle. And that is they function through the sodium movement of sodium-potassium. Sodium is pretty much outside of the cell. Potassium is pretty much inside of the cell. During depolarization, the sodium-potassium pump kicks in and moves the sodium inside of the cell and the potassium out. Calcium rushes into the cellular environment during this time frame and elongates the contraction. Whenever the repolarization occurs, the body tries to reset itself or go back to a resting state. So now the sodium that's inside the cell is moved to the outside environment, and the potassium that is outside the cell is moved back into the inside environment. And that will be the completion of Chapter 3, Cardiac Electrophysiology.